Okay, I'm starting. Thank you all of you to give this precious opportunity to present the paper. So today I'm going to present the paper, famous paper by W. Liu and D. Walker, which was published in Science in 2013. So the topic is serial femtosecond crystallography of G protein coupled receptors. So outline of my talk will be, I have some slides for G protein coupled receptor and its importance, and then after I have some slides for LCP crystallization method. So I will talk about serial femtosecond crystallography at XFEL, and in this paper the authors are mainly comparing the results from XFEL versus synchrotron data. So I will show the results and conclusions. So G protein coupled receptor, why it is important and what is it? So G protein coupled receptors represent a highly diverse superfamily of membrane proteins that mediate cellular communication. So about 800 GPCR respond to a variety of signaling molecules and transmit signals inside the cell in humans by coupling to the G protein and other effectors. So this receptor involves in a key physiological and sensory processes in humans, which makes GPCRs as a prominent drug targets. So still the knowledge of the structural mechanism of ligand recognition, receptor activation and signaling in this broad family is unknown. So there are a lot of challenges involved for these structural studies, including low expression yields, low receptor stability after detergent extraction from native membranes, and high conformational heterogeneity. So far only 22 receptors structures determination is done, some of which were solved in several conformational states and one in complex with its G protein partner. So this is the GPCR, and here the ligand named as ergotamine is attached with it. So its study is very important, and so far 8 Nobel Prizes have been awarded for some aspect of G protein mediated signaling, and the last Nobel Prize awarded was in 2012 for chemistry. So I'll talk about LCP crystallization. So they use LCP crystallization technique to obtain the crystal for this receptor. So LCP crystallization, it is proven as successful for obtaining high resolution structures of a variety of membrane protein, including other ion channels as well as these GPCRs. This crystallization method is not difficult but requires an understanding of the lipid phase behavior and practice in handling viscous mesophase materials. This method leads to highly ordered crystal, but they are very limited in size. So here is the crystal nucleation and growth. So the figure right side down is showing that crystal growth, and the figure on right side, this A, B, C, and D, it's showing optimization trials to improve the crystal growth. And the figure on right, A and B, these are the bright field and cross-polarized images of crystals used to obtain the synchrotron data, and C and D, these are the corresponding images of crystals to obtain XFEL data. So let's talk about XFEL. So XFEL pulses, we know they are 10 to the power 12 times stronger than the strongest synchrotron. So XFEL pulse is used to minimize the radiation damage as well as high resolution room temperature structure of human serotonin receptor using sub 10 micrometer microcrystal is obtained. So this is the LCLS facility where we produce the XFEL beam. So now I have some slides about serial femtosecond crystallography. So in this method, you just inject the sequence of particles which interact with the XFEL beam, and the corresponding diffraction pattern is recorded in the pixel detector. So the highly intense and ultra-short X-ray pulses produced by XFELs enable recording high-resolution diffraction snapshots from individual crystal at single orientation before their destruction. So SFX data collection therefore relies on a continuous supply of small crystals intersecting the XFEL beam in a random orientation, typically provided by fast-running liquid microjet. So for many membrane proteins, including most human membrane proteins, obtaining such quantities is not practical. So here is the experimental setup. So they extrude the crystal through the LCP injector, which interact with the XFEL beam, and that XFEL beam is focused by KV mirrors, and the diffraction patterns are obtained. So the room temperature GPCR structure is obtained at 2.8 angstrom resolution using only 300 microgram of protein crystallized in LCP. So SFX experiments were performed at the CXI instrument of the LCLS. So LCP grown microcrystal, average size 125 micrometer cube of the human serotonin receptor. So the representation is 5HT2B, which is bound to the agonist ergotamine, where continuously delivered across a 1.5 micrometer diameter XFEL beam using a specially designed LCP injector. So these are the diffraction shot. Single pulse diffraction patterns were recorded using 9.5 keV, 1.3 angstrom X-ray pulses of 50 femtosecond duration at a 120 Hz repetition rate by a CSPAT position at a distance of 100 millimeter from the sample. So total of 4,217,508 diffraction patterns were collected within 10 hours using 100 microliter of crystal loaded LCP corresponding to about 0.3 milligram of protein. So of these patterns, 152,651 were identified as the crystal hits corresponding to around 15 crack peaks by the processing software Chitaha, corresponding to a hit rate of 3.6%. So of these crystal hits, 32,819 patterns were successfully indexed and integrated by CRYSTFEL at 2.8 angstrom resolution. The structure was determined by molecular replacement and refined to RWOC around 22.7%. So here are the results. So the significant result is they are able to get the electron density in almost all residues. So this is the, in figure A represents the density map for the helix 5 of the receptor. Figure B, the right side figure B is corresponding density map for the ligand ergotamine, which is attached with the receptor. And figure C is for the simulated annealing ligand density, omid density map. So they are claiming that the final structure has a well-defined density for most residues, including the ligand ergotamine. So it can be claimed that you can get superior quality of electron density from XFEL data, and we can say goodbye freezing and radiation damage which occurs from synchrotron sources. So in that paper, the XFEL structure of this serotonin receptor ergotamine complex, it is compared to the recently published structure of the same receptor ligand complex obtained by the traditional microcrystallography at a synchrotron source. Synchrotron data were collected at 100 Kelvin on cryocooled crystals, which has much larger size. Average volume is near about 10 to the power 4 micrometer cube. And the data collection protocols are listed in table, which I will show later. Both data sets are processed in the same space group C221, which is expected to give the very similar crystallization condition. So these are the tables, and they are comparing the corresponding XFEL data with the synchrotron. So here they are doing this experiment at room temperature. In synchrotron it's 100 Kelvin, and then wavelength is almost same. Beam size, there is considerable different, and completeness, here you got 100%, there you got 90.5. So I will show this correlation plot later, and there are significant improvement from the XFEL data as compared to the synchrotron data. So maybe I will let you some seconds so that you can see these parameters compare. So this is the Pearson correlation coefficient value versus resolution for XFEL data and synchrotron data. So in higher resolution, it's almost 1, but there is considerable difference in in lower resolution, there is almost same, but in higher resolution, there is considerable different. So the lattice parameter for the room temperature XFEL crystals are slightly longer in the A and B direction and slightly shorter in the C directions, which results 2.1% larger unit cell volume. So because of that larger unit cell volume, there is slightly lattice change. We observe a 2.5 degree rotation change of the Brill fusion domain with respect to the receptor here. And these are the main results. So from XFEL data, they are able to recover the electron density for the GLU 212 side chain. They are showing this electron density for the GLU 212 side chain is fully recovered from XFEL data. However, that is not possible from the synchrotron data. And here in B, there is a salt bridge between GLU 319 and lysine 247, links the helices 5 and 6 in the XFEL structure. However, from the synchrotron data, in synchrotron data, it is lysine 247 makes a hydrogen bond with tyrosine 1105 from the Brill fusion protein. So this is the prominent difference they got. And here from XFEL data, the extracellular tip of helix 2 forms a regular helix in the XFEL structure, where from synchrotron data, water stabilized kink is introduced at that position. And in D, from XFEL data, it is seen tyrosine 87 forms a hydrogen bond with arsenine 90 in the XFEL structure, whereas in synchrotron data hydrogen bond is broken and tyrosine 87 adopts a different rotamer conformation. So in that paper, they are talking about this B factor. So B factor normally gives the disorderness from the main position. So these B factor values can be affected by errors associated with the experimental conditions. Their distribution generally represents the relative static and dynamic flexibility of the protein in the crystal. So both structures were obtained from similar samples, and at similar resolution, analysis of their B factor distribution is done to study the effect of the different temperature on the thermal motion of the receptor. So this is the result. The average B factor for the receptor part in the room temperature XFEL structure, they got 88.4 angstrom square, which is 21 angstrom square larger than the corresponding cryo structure, which is 67.2 angstrom square, and it is consistent with the larger thermal motion at room temperature. And another reason is possible effects of Bragg termination during the XFEL pulse. So this is the plot. Here the loop part of the receptor is showing the considerable B factor. So in plot you can see, so this ECL1, ECL2 part, they are higher than average, and ICL2 is showing the prominent B factor change, whereas ICL1 and ECL3, this lower part, this is showing somehow average, a bit more than average. Other helix, 7 helices, they are not showing prominent change. So that thing I have mentioned there. So another prominent thing they found is this XFEL structure confirms the rigid structure conformation of ECl2, which is stabilized by the comprehensive network of hydrogen bonds involving residues lysine 193, GLU 196, arginine 213, aspirin 216, and a lipid ONC. So no other water molecule is observed, emphasizing that water is more disordered and probably doesn't play a substantial role at this location. And this is the another result they showed here. Several side chains are missing electron density in both room temperature and cryostructure. Such lack of density is likely related to the disorder of the corresponding side chains. So here, if corresponding to the XFEL structure, in most of the residues, they are getting the electron density, but in other side chains they are still not getting, which is far better than the synchrotron.
What does CB and CG mean here?
Oh, CB, it's the so once you have that carbon chain, it is linked. The residue is linked with that carbon chain. So CB is your beta carbon. So the carbon immediately after the alpha carbon, and it corresponds to the residue.
So you're saying,
and they are not.
You only have 2 carbons.
Yeah, so it can go up to higher also. So like alpha, beta, gamma, it goes like A, B, G, D, like that. Can I continue?
Yes.
Okay. So here from XFEL data, they are mostly recovering. At least several side chain are still missing. So this is the another result. These 2 disulfide bond, cis 128 and cis 207 and cis 353, they are intact and well resolved in both structures. And here they found the corresponding that beta factor, which is even less than the average because it is intact. So they measure from synchrotron structure, it is 11.1 angstrom square, and from XFEL they measured 11.1 angstrom square, and from synchrotron it was 4.7 angstrom square, which is lower than the average factor increase. And they shown these results, which shows that there is some kind of interaction between the charge residues occur. So they are saying this interaction between charge residues appear stronger and better defined in the XFEL structure compared to the synchrotron structure. So here you see here the corresponding distance is 2.65. This cursor is not working. 2.65, and here in the synchrotron it is 4.15 angstrom. So maybe this is due to this strengthening of the charge interaction is due to the reduction of dielectric constant of water at high temperature, which reduced the dissolution. So the conclusions are, so whatever they observe, they attributed those observations to the thermal motion, and then there is no cryocooling and radiation damage. So this XFEL source enables access to a room temperature GPCR structure, which more accurately represents the conformational ensemble for the receptor under native condition. And since dynamics are the integral part of GPCR biology, use of serial femtosecond crystallography to accurately determine GPCR structure details at room temperature can make an important contribution to understanding the structure function relationships in this GPCR superfamily. So these are the conclusions. So thank you all of you for your attention. If you have any question, I'd like to answer. And this is the acknowledgements. So I would like to thank all of our UWM professors and colleagues, and this material is supported by STC program and National Science Foundation grant number, corresponding grant number.
Thanks, Mark. Any questions?